While I'm here, just one thing. My name is Leo Chan from Massachusetts General Hospital and Harvard Medical School. I co-organized this with Bob Powders, so he is there as speaker too. One thing I want to mention is this is while I'm chairing this a session like this, I always have one thing to say. If you are a graduate student or postdoc, if you ask a question afterwards, come here, I give you a free gift. Okay? So while we're waiting, so this is NMR workshop. If you see the title, I'm pretty sure everyone come here knows this. NMR is a technology which has been, as we all know, about 70 years by now, probably. And then with the metabolomics, NMR is a kind of, we have been there all the time, but not as much as mass spec and other technology which currently dominate that. One reason probably you will hear from the panel is the expertise, the instrumentation, which is much more demanded than running mass spec, as far as I'm concerned, because I don't know too much about mass spec. So because of that, and then we're trying to see what is the thing which we can do to actually enlarge the field in addition to the mass spec, whatever we're seeing right now. So do we have the first slides? Yes, so this is actually just the introduction, which is which is basically a description you can find from the program. Can we have the next one? Yeah, I can do that. Excellent. So we're very fortunate we have 5 distinguished speakers today. Each one will cover a topic which is important to NMR metabolomics. With that, our first speaker is Dr. Art Edison from University of Georgia, and he will discuss about carbon-13 probes and high field NMR. And forgive me, and Art got his PhD in protein NMR with University of Wisconsin Medicine, and he also did postdoc there as a zoology in the Wisconsin Medicine. Afterwards, he has been moving from assistant professor to full professor of University of Florida and the high field magnet lab from 1996 to 2015. Currently, he is a professor at University of Georgia and also director of NMR facility in the Complex Carbohydrate Research Center. So Art, please.
Thanks, Leo. Thank you, Leo and Bob, for organizing this, and thanks everybody for coming out on this first day. It's nice to see everybody here. I'm going to tell you, well, you know, a lot of times NMR people probably we don't do ourselves a great service by advertising the fact that we have low sensitivity. We have low sensitivity because it's really not that bad, but it's complicated. I'm going to give you a little bit of the backstory behind that. And the first thing I'm going to introduce is a facility that we have that is funded, that we're about halfway through a grant where we're building this thing. And I just want to make it clear to everybody, especially in the U.S. and North America, that you can come to our facility if you want access to some of the high field instruments that I'm going to be referring to. Europe has had several gigahertz class instruments put into their facility in Europe, and now we're at the point where we're starting to put our first ones here. So we have a partnership with the University of Wisconsin at Madison and the University of Connecticut And we at UGA, we're putting in a 1.1 gigahertz NMR funded by the National Science Foundation that will do solution state structural biology and metabolomics. Madison's going to focus on solid state structural biology and material science. And then importantly, all of the data are actually integrated in UConn through and securely, so that basically there's a great data management and a really good platform to do data analysis called NMR Box. So that's what we're utilizing. So essentially, we're trying to make this discoverable. So if you don't have a lot of expertise in NMR, but you think that you have a project that could benefit it, we hope that you can go into a portal. You can discover where you could find resources, the best kind of technology that could help you. And then all of your data will go and get securely stored and backed up in the process. We're developing 4 different knowledge bases, we call them. And the 2 knowledge bases that are at University of Georgia are solution structural biology and metabolomics. And so we're really trying to come up with some recommended standard protocols, some analysis pipelines, and we'll be happy to let you access these instruments. You can do it remotely if you send samples and utilize them. So we're going to get delivery of these instruments sometime in the rest of this sometime before the calendar year 2023 is up. But in the meantime, we have some big instrumentation at our facilities that are available. You can reach out if you would like to try to access them. So why do we use high fields? I guess that's a real fundamental question. You know, a lot of places have 500s and 600s. And the 1.1 gigahertz or 1.2 gigahertz instruments are much more expensive. So why go to that expense? Well, I'm plotting here curves that show the theoretical gain in sensitivity. So signal to noise ratio is plotted on the vertical axis, and horizontal axis are field strength. Everything is normalized to 600. So 600 megahertz is the origin, and the blue curve at the top is what happens if the signal to noise scales as omega, which is the frequency, to the 7/4 power. You see that around a lot. And then there's another relationship below that is the signal to noise ratio scaled to the 3/2 power. You also see that a lot. And then the final curve is actually what if the signal noise goes up linearly with the field strength. So a lot of times we get very optimistic and think, well, 7/4, why not? That sounds great. And maybe that will work here. Turns out that 7/4 really only applies to room temperature probes. It doesn't apply to cryoprobes, which most of us use right now. And 3/2, omega to the 3/2 is the best we can do really with cryoprobe technology. And incidentally, I'm going to give a poster number that I can explain a lot more to you if you want to come to a poster during the regular meeting, and I'll explain this in more detail. But the real question then is, how does this do experimentally? So I'm going to show you some data comparing 2 reference standards that we have. One is ethylbenzene and chloroform. It's a non-lossy solvent. And then sucrose. These are both standards that are used for signal to noise measurements. And I'm going to show you data that were provided to me from St. Jude's Medical Center. They have a 1.1 gigahertz installed. And unfortunately, it's not all that impressive. So their data are scaled according to a measurement the same standards measured on 600 that we have. And actually, both of these measurements at 5 millimeter tubes, which is what we oftentimes use, are performing worse than just a linear relationship. So much worse than omega to the 3/2. If you put them into a smaller tube, which is often something that we do to try to alleviate some of the problems that are there, it improves, but it doesn't improve as much as it should. So this is a real problem, and I just want to put a plug in for this poster, because this poster, 1426, I really kind of unravel the basis for this, and there's some pretty simple explanation for why this is the case. The bottom line for here is that The sample is the problem. The electronics, the magnet, everything is about as optimal as it can be, but the sample itself is the problem with the degradation of signal to noise at these high fields. And again, I'll explain this at my poster if you want to come and look. What can we do to try to fix that? We can live with it and just keep on saying, well, we don't have great signal to noise. We can stop using cryoprobes. If we stop using cryoprobes, the sample effects are lessened, but they don't go away, especially at higher fields. And in fact, but everything else is lower sensitivity, so it's not a great solution to stop using cryoprobes. Really, I think what needs to happen is that we need to work more to optimize sample preparation. And one of the solutions that I'm going to address here is we can detect other nuclei. We don't have to detect proton. And in fact, as field strengths become higher and higher, other nuclei are going to become much more important because they have lower frequencies. So I'm going to talk for the rest of the time about C13 detection in metabolomics. And this is a field that probably many of you are not familiar with. We normally think of NMR as we detect protons. Even if we do 2D experiments like HSQC, that's detecting protons. You indirectly detect C13. But C13 detection actually has some big advantages that I'll try to tell you about. First of all, most metabolites have carbon atoms. Can't see everything. We can't see phosphate or just ammonia or something like that. But most metabolites have carbon, so that's a good thing. We can actually do useful metabolomics with this. The frequency of C13 is 1/4 of the frequency of proton. So wherever proton hits a problem, carbon still has 4 times as far to go. So before it really has the same sort of sample loss. So sample loss is frequency dependent and sometimes steeply frequency dependent. So there's some big advantages in terms of the effects that I'm showing on that first slide. C13 has a huge spectral window. So if you've looked at it, it has about 200 ppm spectral width. Compare that to 10 ppm for proton. I'll show this with some data on an upcoming slide that you can just see for yourself that there's less overlap for the same species that you have in there. And then even if you don't have, even if you have no attached proton, so again, if you are used to some NMR and you've done an HSQC experiment, heteronuclear single quantum experiment, that relies on a carbon-13 being attached to a proton. And you can, with the C13 detection, you can see quaternary carbons, carbonyls, everything. You get very narrow singlets for natural abundance C13, which really helps so that you don't have J coupling that are interfering with the spectra. And you don't have any solvent to worry about. So C13 detection is really lovely in terms of you just don't have to worry about what kind of solvent suppression you're going to do. The problem is, and I think that we would all be doing C13 NMR if it were more sensitive than it is. So that 1/4 frequency that it has compared to proton also translates to lower sensitivity, intrinsic sensitivity, because sensitivity is also frequency dependent. But at natural abundance, it's also the concentration of your sample is 100-fold less than proton for the same thing. So it's tough, and this is why it's not routinely done. There are options that are available right now. Maybe in many of your labs you have some. So I'm going to show you what is out there right now commercially, and some of them are quite good, some of them are not so good. And I'm just going to flip through 3 different commercial type probes that you may or may not be familiar with. And I'm going to put values called ASTM. These are measurements that we use to calibrate the signal to noise of C13 measurements across different probes. So many of you are probably most familiar, probably most of you, if you have any experience with NMR, have come across a broadband probe. And these are room temperature probes. You can tune the frequency to any kind of X nucleus. In this case, if you tune it to C13, you can get a nice spectrum, but at 11 Tesla, which is 500 megahertz, you would get an ASTM value of about 390, which just park that in your brain for the next bit. So these are really common, but it's really too low to do a lot with. Bruker actually makes two different cryoprobes that are good for carbon-13 measurements. One is called a TXO, X for either C13 or N15, and O is for observe, and they're cryoprobes. And we have one of these on our 900, which is 21 Tesla, and at that, for that instrument, it's about 3,000 or a little bit more than 3,000 for ASTM. So almost, but not quite 10 times better than the broadband at room temperature. And these are all a little bit apples and oranges. They're different field strengths, but they're ballpark. And then there's another probe that Bruker makes called a DCA cryoprobe, and it has about that same 3,000 ASTM value. In this case, it's 600 megahertz, and that just is optimized to detect C13. So I'm putting these out there because if you really want to get into this and you want to invest the money in either one of these cryoprobes, they're not cheap, but they're about the same price as a conventional cryoprobe. And so they're available, but they're also available through NAN in our facility right now. So if you really want to try this out, we have those last two, the TXO and DCA running, and we'd be happy to try out a sample for you. There's an alternative, and this is what I'm going to tell you about the remainder of the time. I've been involved for about 15 or 20 years now with people at the Magnet Lab, building specialized probes made out of high-temperature superconducting material. These things are, and they always involve a commercial vendor. I'm just highlighting one of our projects that was published several years ago for 600. We no longer are really using this, but it shows the complexity. So you can see the drawing at the top that these are actually flat wafers that surround a sample and give really high sensitivity. So this probe actually had just a 1.5 millimeter sample, so around 35 microliter sample volume, so very small probe. If you make them smaller, they have more mass sensitivity. But one of the points that I want to show was from this initial publication, where if the top spectra are C13 measurements, natural abundance of a mixture of compounds that we put in. The bottom spectra with an expansion is the same sample proton detection. And the bottom line here is that when you look at the top, there's more noise, but there's more space between the peaks. So if you want to do multivariate analysis and statistics, you've got a lot more space. You have a lot less overlap in C13 spectra, which is very compelling. You can also, if you can make a sample that's C13 labeled, it's really impractical to do this if you don't C13 label, but if you do have a way to get a C13 labeled sample, you can do an experiment called Inadequate, which is a carbon-carbon correlation map, and that actually is really efficient with the kind of technology that I'm talking about. We had a paper just a few months ago, Mario Uchimiya, and he has a poster, number 1451, where he's going to present this kind of technology using mouse samples. The paper that I'm showing you here, the data are from phytoplankton that were grown with C13. So anything that you can label with C13, you can do it. Mouse is a lot harder. But on the bottom is an inadequate spectrum from a metabolomic sample from phytoplankton. And then Mario developed C13 J-resolved spectroscopy, so you can actually get rid of the multiplets that you add by putting in carbon labeling, and then you can make a projection and do multivariate analysis with that. So now I'm just in the final couple of minutes, I'm going to tell you about a new probe that is almost ready to be commissioned in our facility and will be available if you're interested. The problem with the probe that I just highlighted is has great mass sensitivity, but a very small volume, and so you still don't have a lot of. It's hard to do it's hard to do a sample like human urine or something where you're not limited in sample quantity, but you can't really concentrate it in there too well. So we built a probe. We designed a probe. This is an NIH-funded project that took several years, and we're just finishing it up now. And it has an unusual geometry. So if you look on the right-hand side, it shows the cross-section of what this looks like. It actually uses a sample tube that's 3 by 6 millimeter in diameter, so it's a rectangular sample tube. We wanted to get as much sample facing these carbon coils, which are the flat structures on both sides of that inner sample changer. And then you can see a cross-section of this thing with the carbon coils or the flat structures, and then around the outside are proton coils. So this takes a ton of work. And so it was actually literally 2 to 3 years of design and simulation. I'm not going to tell you details about that, except just to show that it really takes a lot of effort to put these together to make these things. I'll be happy to talk about this with you separately. But and this is just showing what the tubes look like. They're rectangular. We have a special spinner. It self-centers itself so it doesn't break when it goes down in the wrong orientation. The bottom line with this is it's a remarkably good probe. If you have your sample in a low loss solvent, like in this case chloroform. So this is a sample of 20 millimolar cholesterol dissolved in chloroform, and this is an inadequate spectrum at natural abundance carbon 13, and it took around 24 hours. So we could easily, this has great signal to noise, and we could easily take this down to say 10 millimolar natural abundance carbon. The carbon 1D spectrum at the top was actually collected in about 3 minutes. This probe, if you remember the numbers that I gave you for ASTM earlier, around 3,000 for the best probes that you can buy commercially, this has an ASTM value of 5,500 roughly. I say roughly because it changes depending on a lot of different conditions, but with a smaller sample volume, 425 microliters. So it's really a sensitive probe. The problem is that because it is so sensitive, it's also experiencing these loss problems that I described in the first slide for proton. And so the origin here is a sample of glucose at 25 millimolar, and we're just calling that 1. Or we're starting with the signal to noise, and if you put in, the blue curve is titrating in sodium chloride, which really degrades the signal substantially. The upper curve is titrating in tris buffer, and it doesn't degrade the signal the same way at the same concentrations. And again, I can explain that to you in my poster 1426 if you want to find out why. So what can we do besides chloroform? Well, we can actually detect, this is a human urine sample. The upper, so this is in D2O. The upper spectrum, the carbon spectrum took 4 hours to collect, so it's not a fast experiment. It's natural abundance. We didn't get C13 labeled urine. But we can actually get usable spectra after 1 hour. So I think that for screening, we would probably do 1 hour runs with human urine. The spectrum on the 2D spectrum on the bottom is a het core. And this is sort of an old-fashioned HSQC. NMR people used to use het core. It was carbon 13 detect, but then HSQC came along. It was more sensitive, but het core actually has a lot of advantages because the high resolution axis is now C13, and it's in the acquisition, and so we can get really high resolution data. But you can see there's a lot of richness in this HSQC spectrum that we can get from this, and this is going to be available soon through NAN that I've talked about at the very start. So in summary, high field NMR has a lot of promise, but in order to get the full benefit, we really need to pay a lot of attention to samples, and I don't think we've done that. We as a community haven't really figured this out totally yet. C13 does have big advantages, and that will only continue as field strengths increase. And then you can get increased resolution, you can get C13 correlations. If you have a labeled sample, there's less salt and pH-dependent effects, and it's a great nucleus if you're going to compute chemical shifts with quantum chemistry. So that's it. Thanks especially to the people at Magnet Lab and Brooker for all of the help, and then my colleagues in NAN. And thanks to all of you.
Any question for Art?
So we're going to do question answer. Yeah.
Why don't we do this? If anyone has very burning question, we just ask now. If you want to later think about, and then with panel is still here.
Sounds
that makes
great.
sense? Anyone? Any burning? No? No burning? Okay.
Can I ask one? Sorry.
Yeah,
So-
go ahead.
Yeah.
Go ahead.
So the rectangular tube, is that to solve a sample requirement or some added benefit in like the detector and collection or both?
Okay, so it's a complicated answer to a simple question. The bottom line is it's a way to put more material in with the carbon coils closer together, and so we're packing more sample between the carbon coils. The problem is that we've created a bigger problem with the salt effect because the electric field is now going along the long axis of that sample rather than the short axis. So if we took that same sample and turned it perpendicular to the carbon coils, we would see far less effects, smaller effects on like degradation with sodium chloride, but we'd have smaller absolute signal to noise. So it was sort of a trade-off. We knew that we'd see it, but it also, if we put proton coils in there that would be sensitive. Right now the proton is far away, we wouldn't have such an effect with those. Yeah. But it's basically to get a lot of sample in there.
Excellent. Any other questions? There's a microphone here if you want to. If not, maybe we move on for now. Our next speaker will be Dr. Goncalo Gouveia. He is from University of Maryland and the National Institute of Standard and Technology. He got his PhD from University of Georgia with art. And he did a postdoc, right now is a postdoc scientist at University of Maryland and the NAST. His topic will be applying the known to unknown, a long-term framework for metabolite ID. Goncalo. 18 minutes.
Yes. So first and foremost, I'm sorry for not being Lloyd Sumner, because he's the one that should be here presenting today. So I'm kind of taking on his role a little bit. So if you're interested in the intersection of NMR and LC-MS, I might have something that might pique your interest. So today I'm going to talk about work that I started as a PhD student in the arts lab. And basically when we started this work was really to find a way that we could develop a pipeline or a framework that we could identify metabolites that were not present in databases. And also to use that same pipeline to validate database matches that you would get from your studies. And as you know, metabolomics compound identification is very hard, and that's mainly because we try to measure a lot of things that come from many different sources. And those metabolites that we're trying to measure, they have very different physicochemical properties, so they're hard to find a method that fits all. But at the same time, we want to be able to identify and isolate two compounds that are in the same class and are very, very, very similar to one another. And that makes it a very interesting analytical problem, or analytical chemistry problem. And we can see that when we look at our spectra, right? They're busy, they're noisy, there's varying degrees of signal to noise depending on the metabolite you choose. And for that reason, we try to use statistical analysis, computational methods to transform our data and kind of squeeze out the features that are interesting for our biological study and our hypothesis. But really the critical part of the metabolomics workflow is really to identify that metabolite, because that's the only way that you can give biological meaning to the analytical output of your instrument. And if you get that wrong, then your interpretation of your system is wrong, and it's not cool. So basically, how do we do this identification? The best way to do it is to compare it against an authentic standard. For NMR, you can spike your standard into your sample, and you see your peaks go up, and that looks great. And you can collect additional 2D data to increase your confidence of that annotation and that match. On the LC-MS side, you need to run your authentic standard alongside your biological samples, and then you can compare your retention time, your molecular weight, and validate that match with a fragmentation pattern. And if you manage to do this, it's great. You have achieved the MSI level 1 compound identification level, which is really as good as you can get. And in fact, this is normally what people do in targeted metabolomics. It's when you know what you want to measure and you want to measure something carefully, use standards to do that identification. But it heavily relies on having those standards, which can get expensive. They add sample preparation time, they add run time, and it's not really compatible with an untargeted approach where you don't really know which metabolites are gonna be interesting until you actually measure and analyze your data. So the approach for that is really to grab all your data, your 2D data, and match it against a database that has hundreds of thousands of spectra from authentic standards. And this is great because it's automated, it's fast, and you get hundreds of metabolites that get identified. But there's some significant limitations to this. And in fact, that's why the Metabolomics Standard Initiative guidelines say that you have to knock it down to a level 2 because your confidence on that annotation is just a little bit lower. So there is really a need then to validate these database matches that we get, because if we don't, then you might be identifying the wrong compound. You might be calling the wrong thing to the wrong metabolite. And also we need to have a pipeline where when we don't have a match against the library, what do we do next? We need to have a path forward that we can identify that metabolite because it's interesting and your data is telling you that it's interesting. So as you can imagine, NMR and LC-MS, they're great tools for this kind of validation because they both measure different properties of the same molecule. So you can basically validate your annotation by using both instruments. And not only that, it generates data that can allow you to draw the chemical structure of an unknown metabolite from scratch. But as you expect, that is not very straightforward. It's quite challenging, mainly because of the sensitivity gap between the two instruments, with LC-MS being, like Art said, far more sensitive than NMR, but also because we're measuring biological samples. So our spectra are noisy and they have a lot of samples, which makes it really hard to measure a metabolite with NMR and grab those same set of features and compare them against that same metabolite on the LC-MS data. But it turns out that the natural products chemistry has kind of figured this out a few decades ago, and they use this process called fractionation. And it essentially consists in grabbing a bunch of material, putting it down an HPLC column, and trying to isolate and separate and concentrate that particular metabolite that has a specific biological activity of interest, whether it's antimicrobial, antiviral, and so on. But as you can imagine, applying this kind of mind frame and this kind of work into metabolomics is not really very compatible, because you'd have to do this for every single metabolite, for every single study that you do, and that's just logistically impossible, if not very, very expensive. And also the amount of material, like I said, fractionation requires a lot of material to start that process. And metabolomics, we normally use or prefer higher number of samples for that statistical significance rather than higher amount of material per sample. So we kind of figured out a way, or realized that there was a way around these metabolomics challenges in the application of the fractionation process, and that was to develop a reference material. Instead of using samples, we'd use a reference material. So large part of my PhD program was really developing a method that we could create a reference material that was stable, that it was logistically friendly, so you didn't have to buy tons of material and buy a new -80 freezer to put all that material in, that would be consistent and stable over the very long period of time. So how does this old tricks or group of old methods put together work? So essentially you generate your reference material, so you have a ton of material. You include the reference material in every single sample that you use, and you can use it as in every single study that you do, and you can use it as a quality control sample. And then when you find a feature of interest that's also present in your reference material, what you can do is then go and look at your fractionated data, select the tube that contains your metabolite of interest, and collect your high-quality structural data to make up that particular metabolite chemical structure. And the beauty of this approach is really that you only have to do it once for one type of sample. So if your lab focuses on C. elegans, like Art Labs does, then we create a reference material that will last us until, well, indefinitely because of the method that we developed. So you really only have to put the effort of fractionation once, and then you have high quality data there that you can query to your satisfaction. So how did we do this? We generated our reference material, our C. elegans reference material, our PET model organism. We extracted it with both polar and non-polar, for both polar and non-polar metabolites, and we injected each one of those extracts 6 times into a semi-prep HPLC with reverse phase and helic chromatography. And the methods for the HPLC were basically upscale from our analytical methods from our LC-MS orbit trap. And then we collected fractions every 30 seconds. Every 30 seconds. And unlike the natural products chemistry approach that you try to isolate one of those blue peaks and concentrate it as much as possible, our goal really was just to simplify those fractions and concentrate as best as we could. And when we look at unfractionated spectra, proton NMR spectra, there on the top left-hand corner, and I highlighted that region there, which is the dreaded sugar region that has multiple overlap of peaks that is very hard to annotate. And what we saw then on the right-hand side, where we have all the different fractions, so every single line is a spectra for every single fraction, we saw that we managed to separate those sugars pretty well. In fact, if you look at the yellow trace below, it's almost identical to an authentic standard for trehalose, which is a typical metabolite in C. elegans. And if you look at the glucose trace, it's also very, very similar to the glucose NMR proton spectrum. So we then looked at the anomeric protons, and it's the zoomed in on the left, and we calculated how much was that concentration, how much more did we concentrate those samples. And we saw that for both glucose and trehalose, we had an increase of 5.7 times more, which is really consistent with the 6 times that we injected our reference material into the HPLC for fractionation. So what did we do next? We took every single fraction that we collected, we split it in half. We took 100 microliters and gave it to Brianna Garcia, my colleague at UGA at the time, and she collected MS1 and MS2 on our orbit trap. And I took the remainder, or most of the sample, to be honest, dried it down, reconstituted in D2O, and analyzed using our 1.7 millimeter probe on a 800 megahertz magnet. And thanks to the sample jet, I didn't have to be there changing every single tube. So we could collect all the 1D protons for every single fraction. And then we collected 2D data for select fractions that we were interested in. So what we have now, it's a very nice data set of both LC-MS and NMR data, but we still need to relate the study samples from our metabolomic studies back to the fraction library, and also relate the features between the NMR fraction library and the MS fraction library. And it turns out for MS, it's super simple. Especially if you analyze the fractions with the exact same method and the same parameter as your study samples, you basically just have to grab your retention time, your molecular weight, and then compare the fragmentation pattern, so you can relate them like that. But it's also interesting that if you draw a straight line like you see in the bottom left corner there, you can get a trace of that metabolite across all the different fractions. And this is because of the way that we collected those fractions. It wasn't a purification, it was just simplifying our spectra. And that trace is almost a mirror image if you look at the same metabolite on the NMR fraction library data. So basically, this is a really powerful way of confirming, for example, an annotation in the MS side with the NMR data. But we can get a little bit more fancy than that. We can grab all our MS2 data, throw it into GNPS, and build a molecular network. And the molecular network is essentially a similarity network where we're comparing MS2 spectra to one another and see how similar or how different they are from one another. And what this does is that we can then go into each one of those nodes, which represents an MSMS spectrum, and color-coded according to which fraction it was detected in. And that way, we can then go back to the NMR data of that particular fraction and confirm the annotation of that metabolite with 2D data. But it also becomes the starting point to identify new metabolites. So for example, you see on the middle right side of that square, there's a node that has no identification, but it's very similar to the surrounding nodes. So it means that we can start picking the structural information of those surrounding nodes and then go back to the NMR and start building the molecular structure of that particular unknown. So it turns out that going from MS to MS in the fraction library is very easy. MS to the NMR in the fraction library is fairly okay, but going from NMR to NMR in the fraction library is really hard because you have multiple peaks that could belong to one metabolite or to many metabolites. So to kind of overcome this issue, we relied on the good old statistical total correlation spectroscopy. So basically, you take all your peaks from your study samples, you make a correlation matrix, and you set a really high threshold, like 0.85 or 0.9. That way, you get all the features that are highly correlated to one another. So in our case, we selected that peak at 0.95, so peak for isoleucine. We got all the features that were highly correlated with that, and then use those as targets to query our NMR fraction library. So essentially, we use Stocksy to get targets, and then we use those targets to query the fraction library. And because of the way that we collected those fractions, there's a structure inside the NMR fraction library, which allows us to use covariance as a pinpoint to where those features are going to be present, and therefore determine which fraction that compound is present. And as you can see at the bottom right-hand corner there, we have the isoleucine spectra in blue, which was maxed at fraction number 41, which was highlighted by that little yellow square in the covariance matrix. So basically today I just told you the experimental side of this project. So how to do the fraction library and how to query the fraction library for features of interest. But there's still ongoing work while we're trying to kind of supercharge this data integration to identify compounds better. And that took inspiration from the Summit approach from the Bruchweiler lab, where you can take the molecular formulas, calculate a number of, or obtain a number of possible chemical structures, and then use the FT calculations to determine what that spectra, that NMR spectra would look like, and then use our covariance tool to figure out and confirm if it's present or not in each fraction. Thank you very much for listening. I welcome any questions you may have.
Any question? Want to use the microphone or just shut up?
Remember the gifts for graduate students.
Remember that, yes. It's right here. It's real. All right, if you want to think about it, oh yeah, please. Do you want to use the microphone? Yeah.
Hi, sorry, I'm new in the topic, but when you go from HPLC to NMR, what about the solubility issues? Because you're probably going from, maybe in my case, aqueous phase to organic, and I have problems with that because if you want to measure that, then how do you deal with that?
Well, that issue comes at the beginning when you do the extracts. So you make the extracts compatible to water, essentially, because D2O is essentially water in terms of solvation ability.
Ah, okay,
So
okay.
when you do the extracts, you make sure that your polar extracts are extra polar so that they can dissolve in water.
Okay, okay.
Any other question? Oh, here, good. All right.
So, sorry about, I'm not from chemistry, so I may be asking stupid question. Forgive me for that. So, on the MNR space to MS space, it's a mapping function at the end of the day you're trying to find with the correlation function which you have got there, right? Did you try to do a more complicated, like machine learning type function approximation for the mapping?
No, not yet. So now we wanted just a simple method that we could query the data directly. So we have that feature. Let's try to figure out how we can find that feature in the fraction library. But you're right. Machine learning approaches would, like I said, would supercharge these kind of queries and probably make them better than just using simple correlations.
Thank you.
Did you experience any matrix effects when you add your reference to your biological sample? For example, I think you gave the C. elegans as example. And typically, when you add these sort of reference samples, you can have these matrix effects. And so have you observed something like this? And what I mean by matrix effects is peak asymmetries, even the baseline can get completely different. If you observe that, how did you handle that kind of situation?
So the reference, and sorry, I wasn't very clear, and you guys, if you want to know more about how to make the reference material, you can grab me during the conference. But the reference material is matrix matched. So we make a reference material using C. elegans. So the same matrix effects that we see in our biological samples, we also will see in our reference material. So it's a live reference material. It's not really a synthetic material. So those matrix effects will be the same, for better or for worse.
Any other question? If not, you can think about it. We'll come back later. Our next speaker will be Dr. David Wishart. David right now is a distinguished university professor at University of Alberta, Department of Biological Science and Department of Computing Science. He got his PhD and postdoc from Yale. And the most place when you see David is if you go to human metabolome database and you look at the NMR data, that's all from his lab, most of them. Okay, David, please.
Thanks a lot, Leo. So it's a treat to be here, and I want to thank Leo and Bob for organizing this and inviting me to present to you. So yeah, I'm going to be talking about machine learning and NMR metabolomics, and I think many of you probably have heard of machine learning, but those who haven't or are looking for a really simple explanation of it, machine learning is different than traditional programming. In programming, we typically write algorithms to do things like addition, multiplication, or series of additions and multiplications. So we have set rules, set structures. Everything is usually constructed either as mathematical or logic operations. So traditional calculators, traditional programs can do things like addition and multiplication. But in machine learning, what you do is you don't build an algorithm necessarily, use an existing model, and you give the model a whole bunch of data. And so in this case, we've given this computer a whole bunch of addition tables that give it some numbers and results, and it may not even know what addition is. It may not even have the algorithm or an adder in the computer. But using the machine learning model, it develops a model for addition. And you can apply it to create models for all kinds of things. And obviously, people are developing machine learning to predict or estimate your preferences for movies or to identify if your credit card has been stolen. Again, looking for patterns and then using that to infer or develop its own program. Now, over the last 10 years, there's been a bit of a revolution in machine learning. We've gone from traditional simple machine learning to something called deep learning. Machine learning is often described in terms of things like neural networks, and these were modeled after very simple brains, say insect brains. Deep learning is modeled after human or mammalian brains, which has, each of them have very thick cortexes or cortices. They have lots of layers of neurons, and that's a characteristic of a deep learning architecture. So in the old days for machine learning, what we would do is we'd take our data, we'd go through it manually and look for features, and that's called feature mining or feature selection. We'd apply it to a fairly simple neural net or some related model, and then we'd get our result. With deep learning, you just have to have your data. It'll figure out the features, it'll also figure out the patterns. And so deep learning is what's made ChatGPT so good and maybe also so scary. It's just using piles and piles of text data randomly collected from all the texts that you've been sending for the last 10 years. Now, machine learning can be used not only for nefarious purposes, it can also be used for good purposes, like NMR. And so I'm going to give you some examples of how we're using NMR to help in certain areas of NMR metabolomics. And some of these are related first into the area of spectral deconvolution. Then I'll also talk about how NMR and metabolomics and machine learning can be used for spectral prediction. And then I'll show you how deep learning can also be applied to improve spectral sensitivity. So in the case of NMR deconvolution, the standard problem we have with metabolomics data in NMR or in mass spec is there's lots of peaks. And we're trying to annotate these peaks, and they're often overlapping with each other or partially overlapping each other. And what's shown on your left is an example of a mixture, a really simple mixture, and that's at the top, and that's in blue. And then we can decompose that mixture into 3 different compounds, compounds A, B, and C, each of them with characteristic or reference spectra. Now we can do the standard problem is add 3 spectra together to get the top one, but to go from the top one and deconvolve it to the 3 others is called an inverse problem. That's a little harder. What you need for spectral deconvolution is a large database of pure compounds. That's why we've collected spectra and put them into the HMDB, as Leo mentioned. But you also have to use complex pattern recognition. Pattern recognition is something that machine learning is really good at. So are people. And in the early days, deconvolution was done manually. Some of you may have heard of the Conomics software, which does that. Now, for NMR spectra, to make this work effectively for machines, you also have to make sure the spectra look clean. They need to have flat baselines, they have to be referenced properly, they have to have good phasing. To do all of that, humans normally do it, but you can also train a computer through machine learning to do all of those things. So we've been working on that for the last few years, and we've developed a package called MAGMET. That stands for Magnetic Resonance for Metabolomics. So it uses a combination of machine learning and a number of expert-based rules that we've learned through the years to do the pattern matching, the pattern fitting. It has automated phasing. It has automated chemical shift referencing. It has automated water removal, automated baseline correction. It also does the peak deconvolution. It also identifies and quantifies the metabolites all at the same time. So when we run MAGMET on a serum sample, we typically get between 55 and 60 metabolites, and we identify and quantify all of them. And that really represents about 99% coverage of the signals. We've also adapted it not only to serum, but to plasma. We've also done it for fecal water, to CSF. We've done it for wine and beer. Some of you may have attended our wine tasting workshop at MANA last year. And a lot of the data that was analyzed for that wine workshop was done using MAGMET. You can adapt it for milk and other fermentation products. These are examples of what is done or what it looks like with the spectral fitting. It will slide in and identify these peaks. You can see that little left corner. So if initially it's a little off, it'll make the adjustments just like a human will. It fills the space pretty nicely, integrates, gets the right intensity, gets the right width. So that means you can get the right quantitation. As I said, the iterative fitting is critical to that, as is the need for a flat baseline, which is done also by the software. This is an example of the performance, and so we're looking at the top one is serum, and the bottom one is fecal water. So serum, as I say, if it's a normal person or a normal mammal, 55 to 60 metabolites are identifiable. This one took a little under 5 minutes. And you can also see the fit. There's a red line and a black line. You have to look really closely because they pretty much superimpose. Again, that's just a measure of the quality of the fit. Below that is another one with fecal water. That's more complicated. And this is typically around 70. It can be as high as 80. It can be as low as 50 metabolites. It's much more variable. But again, fit is excellent as you look at the blow up. Also, the quantitation is very, very good. Often we've got CVs of less than 10%, in many cases down to 5% for most metabolite concentrations. We've been doing this for a while, and we've tried to get people interested in using the software, using the techniques. We find that no one ever reads the instructions. So what we've done is we've tried to make kits. Sometimes people are better at following kits. So I'm with the Metabolomics Innovation Center, or TMIC, and what we've done with the kits is we've put things into batches, we've given the reagents, we've given the instructions, we've given the software. We've also added extra software so you can do some interactive spectral editing, because some people don't like the fits. And that's often for people who've been doing this through tools like Genomics. And typically, because we've also modified it to allow you to detect inborn errors in metabolism, so you can get some pretty interesting compounds in serum. So there's up to almost 100 compounds that can be detected in serum, but 60 is the norm, and it also does fecal water. The kits have been out there for about 6 months now, and it's pretty cheap. It's about $10 per sample. If you want to learn more, you can contact Rupa Mandel, who's here, and she's probably looking shocked because I put her name on there. But again, I think this is an example of how machine learning can really help with identification and quantification in NMR and making things really simple, really fast. Now, we heard earlier about the challenges of identifying novel metabolites in NMR, and often we do the similar sort of thing in mass spec. Is if we can predict the mass spec, we can have a pretty good idea what the compound is. So if we can predict the molecular formula, or if we can map the predicted spectra to the observed spectrum, it helps. So if we're trying to identify novel compounds by NMR, it should help if we could also predict spectra. So the normal route to predicting NMR spectra is to use density functional theory, or DFT, or quantum calculations. I've been a big advocate of using semi-classical methods, and we've used that for a long, long time, and they're roughly about the same. DFT is more elegant, semi-classical is a little easier to understand. But ultimately, what you're trying to do is not only predict the chemical shifts, you also want to predict the coupling constants and the intensities. And so what you're trying to do is go from the compound to what we call a spin matrix. That's the thing that's in the upper corner. And then from a spin matrix, then you can create the metabolite spectrum. So you need to predict chemical shifts and J-couplings and intensities. So you can use machine learning, and people have actually used machine learning to do chemical shift prediction, but not deep learning. So we've started using deep learning. To do deep learning, you need a lot of data, and so what we're actually using is the chemical shifts predicted through DFT. We really tried using chemical shifts predicted, or actually identified, and published in the literature, but the error rate is incredibly high. Roughly 10% of all published chemical shift assignments are wrong. So by using correct assignments, even though they're inaccurate because they're DFT calculated, we can actually train the system a little more completely. So we use these DFT predicted shifts to train the system. And then we took a smaller subset of known assigned chemical shifts from the HMDB where we knew they were absolutely correct. We verified they were correct. We checked double and triple times. And it's a smaller set. And we used a technique called transfer learning. So we learned from the large set and we transferred to the correct set. But that was sufficient to do chemical shift prediction with some surprising results. So this is proton chemical shift prediction. We did this in water, but you can do it for chloroform or other solvents. So on the right is NMR shift DB2 with the performance of the correlation coefficients and mean absolute error. And typically those are about 0.2 ppm. DFT methods are usually about 0.3 ppm in terms of the error. MNOVA, which is software that some of you may have heard of, it's commercial, it has a pretty good system, and I think the correlation coefficients are high, and the error is about 0.15 ppm. The first version of this machine learning method gave an error of 0.12. The latest version is down to 0.1 ppm error. So this is an example. This is quite a significant improvement. It's 3 times better than DFT. It's 2 times better than most of the commercial programs. And at some level, this is basically the error of measurement that we're dealing with. If you look at carbon, again, standard DFT errors between 3 and 3.5 ppm. Some of the better, simpler neural net methods give you errors about 2.8, 2.9 ppm. So that's NMR shift DB and MNOVA. We applied it to first draft. We were able to get down to a 1.98 ppm error. Now we're down to 1.75. There's some outliers, which we've realized were mistaken assignments, and so we expect this to get down to about 1.6 ppm. So we're getting to the stage where chemical shift prediction is almost a solved problem, given the errors that we're talking about here. And this covers not only for water, for chloroform, DMSO, methanol. Now, chemical shift is only one part of the equation. Of course, you need to know coupling constants. And so the same approach is being used now by our group. We're taking a large collection of DFT-predicted J couplings. We develop the model through deep learning, and then we use transfer learning, where we've applied this to a large set of experimentally determined coupling constants, which is also from the HMDB. And again, these have been carefully determined and includes not only positive coupling constants, but also negative coupling constants, which exist. And building that data set has been quite challenging. This is an example of one version of the tool in terms of how well the coupling constants are being predicted. Normally, if you look at things through semi-classical or DFT approaches, they're just all over the map. The correlation's very poor. These were getting very good correlations. So if you can predict with coupling constants and have errors of 0.2 or 0.3 hertz and chemical shift errors of 0.1 ppm, you can kind of do what's shown on the bottom there. So we've got proline as an example. We predicted the chemical shifts. We estimated the coupling constants. But then when we use the deep learning approach, chemical shifts are more accurate. The coupling constants are more complete. And you can see the ML prediction and then the actual spectrum, and they're basically superimposable. So I think deep learning will have a pretty significant impact on how well we can predict spectra and how well we can model them for novel compounds. We're applying these tools to predict every single compound, predict a spectrum at every single frequency for proton and carbon in every compound in HMDB and every compound in the natural product database called NPMRD. Another thing you can do with machine learning is improve sensitivity. This is a little cheaper, I think, than buying a new magnet. But there's a paper published a couple years ago which highlighted how you could take spectra, which are shown from this particular paper. There's a carbon spectrum on the left, both a 2D HSQC and a 1D, and then what you can do with machine learning in terms of signal-to-noise improvement. We were really intrigued with that, and unfortunately, the algorithm has never been published. We don't know where it's really sitting at. But we decided to see if we could reproduce that or improve on it. It was limited to carbon data. We wanted to see if we could do it for proton data. So here we used, again, a deep learning model. It's called an encoder-decoder convolutional neural net. It takes the spectra, input spectra, and sort of reduces the dimensionality, compresses it, and then the decoder part of it reconstructs and expands the compressed spectra. You're doing a variety of things like downsampling. You're modifying the skip connections. You're using sigmoid activation. And we had to play a lot with the different kernel sizes to get things to perform as we expected. So here's a result. So in the top example is, this is 2-hydroxybutyrate. This is a spectrum where we just collected a few scans. I think it was 32 scans. It was a low concentration material. You can see it's very noisy. You can see a couple peaks, but most everything's buried in the noise. Then we use something called Whittaker smoothing, which is basically like line broadening, but it doesn't do that much line broadening. And you can see there's an improvement. We got an improvement about 3-fold in terms of signal. And then we applied machine learning. And you can see the signal goes from 12 to 1 to 2,800 to 1. And I think that's actually an underestimate. The data processing only took a second, so it's not as if we had to acquire. So if we wanted to acquire to get the same signal, it would have taken, I can't even read the numbers from here, about 1 million scans. So it saves a lot of time. This is a close-up of seeing what you see with I guess the sextet and the spectrum, selected region, Whittaker smoothing. You can see it a little bit better. You apply deep learning, and it's basically noiseless. You can extend it to a mixture. So here's a serum spectrum, 57 compounds. Again, it's kind of small to see, but you can see the top one has lots of noise, 2 scans. And then you can see as we increase the number of scans, it took about 2 hours. It looks very similar to what we would have got with the ML processing, but it's actually significantly different. In terms of the signal to noise, in the region we're looking at, we had a 2-to-1 signal to noise, 3-to-1, and then scanning for 1,000 scans, or about 2 hours, we get signal to noise about 70-to-1. The machine learning was able to get signal to noise about 180-to-1, so the equivalent of maybe scanning for another 6 or 7 hours, just using something that we'd only collected for a few scans. So I think the ability to use machine learning to enhance signals is real and potentially very powerful. So it's been used in terms of machine learning around for a long, long time. It's been here with us probably for 70 years. But typically, improvements with old-style machine learning only lead to modest improvements over what you can do through semi-classical methods or DFT methods. When you use deep learning, you get significant improvements, seeing improvements in prediction quality, and sensitivity, and also in spectral analysis. And so my expectation is that as more people apply deep learning to areas like NMR, we'll see some pretty important and significant changes to the field. Hopefully they'll make it easier, make it faster, make NMR much more sensitive to a point that I think it could compete with mass spec in terms of sensitivity. So we've had a number of people, graduate students, postdocs, research associates working on this project, and have been funded by a number of groups. So thank you very much.
Thanks, David. Any questions? We still have a couple minutes here. Oh.
Thanks very much. There was some amazing spectra, David, that you just showed there with the deep learning. Does it get into any problems in a similar way that some of the artificial intelligence algorithms can get into problems? Do you see are there any issues with random peaks being generated and things like that, where, you know, some methods can go through and generate those, and there are some classic stories of people getting random peaks in all sorts of different types of spectra. Do you see that with the deep learning? Is that something you have to unlearn, as it were? Sometimes?
Yes, I mean, the term is hallucination, I guess, typically, where the deep learning hallucinates extra peaks. We have seen a little bit of that, but one of the things that we've been doing is, because the spectral simulations we can do are really, really good now, again, thanks to machine learning, so we can create exact spectra of all the compounds that we're looking at and create mixtures of any type. So we can generate training sets of literally millions of spectra, which none of us would have the time to collect. And these are noiseless spectra. So then we can also train and put all kinds of noise, and we can mess up the spectra in all kinds of ways. And so that gives us a gold standard that allows that. It's much better than what ChatGPT, because there isn't a gold standard for texts or truth, a bottom line truth. So we can create a gold standard bottom line truth. And so I'm not particularly worried that we're going to see hallucinatory peaks or other types of things.
Nice talk, David. For the J-coupling predictions, as you know, it's not trivial experimentally to measure a lot of J-couplings, especially when they're small. And so the question is, like, how if you feed it reference J-couplings that are off, does it just make a whole model of like not quite right J-couplings, or what? Yeah.
So again, what we're doing is we take our measured spectra, and then we fit the spectra with the couplings and predicted couplings initially, and then the predicted shifts, and then we iterate until the fit is basically perfect. And so, I mean, it's possible there could be another combination of couplings, but the spectra are identical to our eyes, and so we're pretty confident that those couplings are correct. And then that's used as the training data set for the couplings. So we can't trust the reported couplings, and we're not using the reported couplings. We're using our own derived couplings.
Thanks, David. We'll have one more question. X-ray.
Probably bouncing off that first question with like the challenges, for dirty samples like urine, how much more involved would the machine learning, I guess, process be needed? Would you need to use for dirty samples or like challenging samples?
So the way that the machine learning works, or the way the system works, is it moves across the spectrum in small windows. So it doesn't look at it holistically. It looks at things and identifies the individual peaks or clusters of peaks, or in some case, peaks on top of peaks. Urine spectra are complicated, but it'll still identify those peaks, even if it didn't expect them there. It'll say, okay, there's supposed to be a peak there. So it isn't looking at a holistic picture and say, this is urine, therefore every urine spectrum must be the same. It just sort of scans along. We're not really sure how it works. I mean, it's something like Whittaker smoothing, but it's just a lot smarter. And so it just seems to pull out the peaks. If you look at an NMR spectrum, and you've been an NMR spectroscopist for a while, you can usually sort of see, yeah, there's a peak here, and there should be a peak, there's a quartet here, but you formally apply line broadening, and it kind of messes it up, but it's just simply saying, well, no, there's a quartet here, I know what a quartet should look like, and it gives you the 1:3 to 3:1 ratio, and it gives you the right line width because it's learned it. So it's, I guess, just inferring what we would do if we just collected more data.
Super quick question. Can you distinguish L and D isomers, like in amino acids and things like that? That can be difficult by mass spec, but also important to know.
As a rule with NMR, I don't think we can. I don't think we see enough differences in, chemical shifts as a rule. I don't know if we can see it in the coupling constants. But I don't know if maybe they're reversed coupling constants, maybe, but I don't think we can as a rule.
Yes, and thank you for the nice talk. I was used to use kinomics, and I was wondering if it's similar, and if yes, what's better?
So you've used kinomics. So kinomics has it's a manually fitting process or a semi-automated fitting process. So what you're seeing with Magnet is totally automated. So we just run it. We press a button and we go home, and the next day all of the samples are processed. So we'll do 100, 150 samples in a day.
All right, so we have to move on. Our next speaker will be Dr. Elizabeth O'Day. Dr. O'Day got his PhD from Harvard as a National Science Foundation awardee. He got his MPhil from Cambridge, and right now she is the CEO and the founder of Olaris, a precision diagnostic company. She has been served on many boards, including the steering committee of Scientific American, and she is also adjunct faculty at the Department of Chemistry in Boston College. Her talk is NMR-based metabolomics, clinical diagnostics. Dr. O'Day.
All right, thank you, Leo. Thank you, Bob, for the invitation. I'm super excited to be here. I think the takeaway from everyone for this session, I hope, is that you're all really jazzed about the power of NMR to do metabolomics. From what we heard so far with this panel, I think improvements in, let's say, hardware from rectangular tubes, to sample processing to ML. We're in an era where metabolomics is really being able to deliver NMR-based metabolomics, wildly new insights, and I think this is going to usher in more innovative diagnostics and more tools to accelerate drug development and improve human health. So before I jump into my talk, just quickly, my disclosures: I am the CEO of Olaris. I'm going to tell you a little bit about our work today. Did I mess this up already? Okay, so quickly, my talk is going to be split into 3 sections. First, just a little bit about what makes a clinical diagnostic and some of the ones that are already in use that are metabolite-based. I'll tell you about our Cerebro platform, which is a nice follow-up from David's talk, where we combine metabolomics and machine learning to develop and discover metabolite biomarkers for disease. And then somebody asked a question about what you do for urine and really messy spectra. That's a lot of the work that we work on. Our goal is to develop sort of non-invasive biomarkers that can improve human health, and I think urine is a really nice input for that, but urine comes with some unique challenges, and we've done a pretty systematic and very non-sexy approach to be able to sort of figure out what is the best way to analyze urine. So briefly, metabolites are great. I don't think I need to explain that to anybody in this room. Altered metabolism is associated with nearly every disease, and thus, not unexpectedly, the world's best clinical diagnostics measure metabolites. You can think of glucose as a readout for diabetes, lipids as a way to predict cardiovascular disease, and even right now, like L-DOPA and response to L-DOPA is one of the only ways that you can determine or perhaps quantify Parkinson's disease. And so metabolites have a well-proven history of being important to make clinical decisions. But to be a real clinical diagnostic, there are criteria that you need to hit. And you have to fit this definition of analytical validity, which shows that you're able to measure whatever the thing is with a defined level of accuracy. It's repeatable, it's reproducible, it's specific and sensitive. And you need to be able to sort of document all those things. And because of some of the complex challenges of metabolites, like a chemical diversity, they're constantly changing, the wide dynamic range, many of the traditional metabolites, metabolite-based diagnostics, although they're great, they're really only ever measuring maybe one or a handful of metabolites to be able to correlate with disease. And I think that means that we are just scratching the surface of what is possible, because it's really being able to measure maybe not the full metabolome, but a more complete picture of metabolite that's going to let us draw even more new insights into disease etiology, human health, biology, and hopefully actually get us to a position where we might move from precision medicine to one of preventative medicine, where we can kind of start to predict what might happen. And so I think, again, this is a really exciting opportunity where advancements in both NMR and mass spec, machine learning, and just our greater understanding of biology, that we're on the precipice of being able to capture more information to develop diagnostics. Again, this is exactly what our company does. And our platform works like this. So we call it the Cerebro platform, and hopefully there might be some X-Men comic book fans in the audience and get the reference. But basically how it works is that we get patient samples in. This could be blood, plasma, serum, or urine. And we isolate all the metabolites. We then will run it through a dual process of doing NMR profiling, where here we do multidimensional NMR. We think the chemical shift over, well, 1D NMR is incredibly powerful. I think the chemical shift overlap, as we've seen in some of the other talks, really prevents doing proper metabolite annotation. And if we really want to know what's going on, we need to figure out what are the metabolites that are present in each patient sample. So we rely on non-uniform samples. We also use a lot of paramagnetic relaxation enhancement in order to get sort of the biggest bang for our buck. For the sake of this talk, I won't probably have time to go into it, but if anyone has any questions, please feel free to follow up with me later. In addition, our samples are also passed through a global panel where we use a LC-MS, a triple quad, where we can detect and quantify right now about 370 metabolites in a single sample. All of this provides, an incredibly rich sandbox for us to do basic statistics as well as really advanced machine learning and AI, where we have developed, new clustering techniques, new normalization, library matching, stats, feature selection, X, Y, and Z, you name it, we do it in the effort to develop this predictive algorithm. One thing that I think is important for anybody that's doing metabolomics data is we all know we get a lot of data, and with a lot of data, and sometimes the world we live in when you don't have this, maybe the appropriate number of samples, you are usually looking at a high dimensionality, low N problem. It's easy to build any model, right? And that's, I think, some of the challenges the field is facing, where David alluded to, like a lot of annotations in metabolomics data is not reproducible. I would say a lot of findings are also not reproducible between one study to the next. If you look at some concordance in different, disease areas, there's very little metabolites that show up as hits. And that's not because any one of those studies is bad. I think actually every single one has been done great for that particular study, but we don't know all the nuances that are there, and often there's not this important sort of vetting step that we call the link to biology, where you actually try to see, do the differences that you're seeing make sense, and can we link this back to, again, underlying biological processes to teach us something more about the disease. So at Olaris, we have 2 clinical products that are in various stages of development that I will briefly tell you about. The first one is a kidney transplant assay, or it's a urine-based assay for kidney transplant patients, where in the discovery part, we got urine from kidney transplant patients who were experiencing different outcomes. So in transplant, these patients have to take immunosuppressants for the rest of their lives so that their immune systems don't kick out the newly formed graft that's been put in their body. Sometimes this can mean that they have to take upwards of like 40 drugs a day. It's really crazy. And it's a challenge for physicians to know what is the appropriate dose for an individual patient. While there are sort of average targets for therapeutic drug monitoring and trough levels, it's really unclear for one patient what's the best way to do it. And this puts the kidney transplant patients in this constant sort of vicious cycle of potentially being under immunosuppressed, where if they're not getting enough of their drug, their immune system can go attack the organ and you get rejection. But if they get too much of these immunosuppressants, it's actually just as bad, and it puts them at risk for opportunistic infections and malignancy. In fact, kidney transplant patients are like 200 times more likely to develop cancer because they're constantly being dosed with these immunosuppressants. So what we did is got roughly 400 urine samples from kidney transplant patients where we had biopsy matched to that sample so that we could use that for our machine learning. We used multidimensional NMR spectroscopy, identified differential resonances between patients that either had a stable graft, either were that moment in time experiencing rejection, or were potentially experiencing an infection. And we used, again, just basic statistics and then cross-validated machine learning models to try to predict, to build a signature based off of metabolites that could help us tell for a single patient what's going on. And I wouldn't be here if it hadn't worked. So you can see here is some data from both our training, and now we're doing blinded validation studies where, based off of the signature of the 9 metabolites that are shown in the violin boxes, we can build an algorithm that near perfectly separates patients who have a life-threatening infection called PVAN, pallomavirus-associated nephropathy, which are they're shown in orange, from people who are experiencing rejection. And this is incredibly helpful because right now, under a microscope, these two things look identical on a pathology slide, or nearly identical, but they have the opposite clinical treatment. And so now from a urine-based sample, we're able to inform or help inform a physician, for a patient that's experiencing a problem, do you actually need to turn up or turn down their therapy? Another one of the products that we're developing is for Parkinson's disease, where I think this goes through all neurodegenerative diseases. Good God, do we need better diagnostics? Parkinson's disease, like other diseases, they develop over a long time, and right now there are no confirmatory diagnostics. So by the time the clinical symptoms manifest, often 60 to 80% of the neurons could already be lost. So really, at that moment, there's not much you can do for a patient. And so is there a way that we could use metabolites, which are sort of kind of like the canary in the coal mine, to be able to identify Parkinson's development at its earliest onset? And so in work that was supported by the Michael J. Fox Foundation and now will be expanded upon, we were able to look at a subset of Parkinson's patients. And I think this is also another powerful utility of metabolomics not shown here. Parkinson's disease is kind of lumped together as one big disease when it's not, right? Just like in oncology, where we used to call everything breast cancer, and now there's like 15 different types of breast cancer. I think we're gonna see that in neuro as well. And so through our work, we've been able to identify plasma-based metabolites that can differentiate this particular group of familial Parkinson's patients, which in this waterfall plot are shown in orange, from age, sex-matched controls shown in blue. And we're pretty excited about this work because, the overall accuracy is about 80%. And Parkinson's right now can only be confirmed post-mortem when you do an autopsy. And it's known that, we have a misdiagnosis of 20 to 30%. So you actually wouldn't want a model to do much better than that because it could be classic overfitting. So we're excited about moving these products forward. And I think through our pipeline, you can point this at any disease area that needs either better diagnosis, better selection of therapy, or better response monitoring. And we have developed this sort of suite of software tools that we're going to be launching soon that we call sort of low-touch metabolomics, that take kind of all the pain in each step and the cumbersome and complex analysis and make it pretty smooth. But what I will say is what I have learned over the 9 years of now running Olaris, while yes, you can do a lot of fancy things on the back end, everything gets said at the beginning, right? And ensuring that you have high quality samples or high quality data, going into your analysis and your algorithms is absolutely critical. And so I'm gonna spend the rest of the talk just briefly talking about some of the things that we've done to give us confidence that the data that's going in is going to give us a robust signature that can have clinical utility. And so anybody that does metabolomics knows there's a lot of different sources of error that you can have. It can come from the sample itself. It can come from the sample prep. It can come in the way that it was collected and all these sort of things that happen before it even kind of gets to the algorithm design part. And so we've been working to sort of develop what we call navigators that can help us just get a better handle of these sources of error and do we need to sort of mitigate them in our analysis. And one of the biggest things that we've realized is in urine, specific gravity is a huge problem. So specific gravity in our case has more than pH, more than salt, more than like anything else, has become the thing that we think introduces most false positives in urine-based analysis. And so again, if I can give a takeaway here, if you're doing urine-based metabolomics, please look at specific gravity. So the first thing that we've been working on is something that we call a sample prep navigator. So because we're doing 2D NMR, we have to concentrate the sample. So we do a Folch extraction, methanol chloroform, and then collect the aqueous layer, and we analyze the lipid layer separately. And you know, a lot of things can happen during that Folch extraction. So we wanted to figure out, is there a molecule that we could add at the very beginning that would let us know that the sample prep went well. And so we pioneered this molecule. It's a proprietary molecule called NAV1 that you can add at the beginning of your sample prep. It does nothing to your spectra, introduces very few differences in the HSQC, but it tells you what happens. So if you do a good prep, either you or like multiple people in your lab, you have a pretty tight coefficient of variation in this particular resonance, you know, less than 10%. But if you mess up, if you don't, you know, extract the right amount of the aqueous layer, or something happened, you know, when you were adding your chloroform, the coefficient of vary and the intensity of this particular resonance changes. And so we use it now, and it's built into our software as a flag, as a first step, like, did the prep work? Should we even trust the data that's going in here? The next thing we do is we use a pH navigator, and this was built off of work that Mike Riley did at Merck, where he developed this molecule DFTMP, which is great, and we've just kind of added on from it. So its chemical shift is very sensitive to pH, and even very small changes in pH. So after we do our sample prep, we add DFMTP before it goes into the NMR, and what we see is that the chemical shift of DFMTP can tell us both the initial pH of the sample before we did the Folch extraction, but also the sample of the actual NMR sample that was run in the magnet. And this is really important, again, when you're dealing with urine, because both we do a lot of biopharma services work, which has a lot of animal urine as well, but the pH of real patient samples and animals is all over the map. Sometimes we get samples that have pH 5, and sometimes I have pH 8, and that initial pH can influence which metabolites actually precipitate, and of course they will influence the chemical shift of your spectra. And DFTMP, we think, helps provide a great readout. I won't talk about this too much because I think Bob might be, because this is actually work from Bob Power's group here, where he profiled a whole series of metabolites and showed, you know, just what we all know, that chemical shift in line with, and everything is a function of pH. And this happens even in very tight windows. So even though that we're using a strong buffer at the end and we get everything to about, you know, roughly pH 7, but there's a little deviations, even those small changes in pH can lead to big changes in chemical shift. And when you're dealing with real patient samples, this can be a problem when you're trying to do feature identification. So this is looking at the histidine resonance, the one marked in orange, in about 100 different kidney transplant patient samples, where if you didn't know the pH of these samples, you might think that each one of these clusters was a different feature, and you might put them into different groups, and then you would say like, oh, you know, the people on the left are missing the cluster on the bottom, and that's not the case, right? Where because of DFMTP, we can actually know what the pH is of each sample and use that to improve our clustering. All right, I will very briefly run through this, but specific gravity, as I said, is a huge potential source of error in doing urinary metabolomics, and I highly suggest people take a look at it. So here we're showing the same pooled urine where we either concentrated it or diluted it to different specific gravities. And if you were just doing sort of standard metabolomics, you would set some signal to noise cutoff and pick your peaks and go forward. That's not helpful, as you'll see, because it will introduce wild amounts of false positives. So let me just quickly show you here. So again, these are those same samples, pooled urine, set to different specific gravities by force concentration or dilution. What you can see just by eye is that in the most dilute samples, you have less peaks, and in the concentrated samples, more peaks and more T1 noise and artifacts like that. So if you just fed this into some automated or semi-automated data analysis program, you would get poop. And so one of the things that we first did was take both a manual and a machine learning approach to see, can we develop tools that are better to help us set spectra to, more consistent data quality. And so what we found is we profiled all different types of parameters to see if you could find a feature that would let you correct for specific gravity post-acquisition. And what we found is that actually, instead of doing a signal to noise cutoff, if you do a sum-based cutoff, you get much more high-quality data. And in fact, you can reduce your false positives from hundreds to practically nothing by just simply changing from the traditional signal to noise-based cutoffs to a sum-based cutoff. You still retain the ability to detect all your true positives. And the thing that I think is really helpful is that it really dramatically improves your ability to do clustering in either an automated fashion or a semi-automated fashion. If you don't correct for specific gravity, you get these sort of unique clusters that will rock across your spectra. But if you correct for specific gravity in the lab, you reduce all of your false positives and you make your spectra beautiful, where all of those chemical shifts go away. So where you no longer really have to spend as much time, annotating every single feature and making sure that it aligns so that you are confident in your statistical analysis. So I know we're over time, so I will end sort of here, which is our proposal for if you're doing urine-based metabolomics to really think about specific gravity. We also tested pH, we tested dication precipitation, we tested DMSO, and by far, bang for your buck comes from specific gravity. Add a pH navigator, add some PREs, and you're gonna get this, which is like a QA/QC matrix, which to me is the first step that we really need before we go on to develop all these super cool and powerful clinical diagnostics. And so with that, I will thank the amazing group at Olaris that is responsible for all of this work. And we're hiring, just to throw that out there.
Great, thank you very much. Any question? We probably have one question, if we have one. Any? Well, no? Not for now. Yeah, okay.
So I was curious about with the specific gravity correction. I agree it's really important for urine, but a lot of people clinically will correct using creatinine. And I don't know if you guys have found that that's not good or whether it's useful or...
It's better than nothing. So correcting to creatinine is certainly better than not, but we, in this paper that we have publishing, showcase that creatinine is not always perfect. So creatinine will change based off of sex, muscle mass, age as well. So while it helps for the hydration status of how well your kidney is potentially filtering out or how much water you've drank, it's not perfect. And so if I had to choose, I'd say total sum and correcting in the lab, but then also looking at creatinine is a good one as well.
Very quick, okay?
Does your machine learning model take the two inputs, pH and specific gravity, as inputs along with your others? Can you bring them into one space?
To include them as features like in a clinical diagnostic?
Not so. Maybe I can take it offline. I think I'm out of time.
Great. Just very quick, okay?
So why not osmolality over specific gravity?
Yeah, so again, we checked all of these sorts of things, and bang for your buck was specific gravity. I'm not sure that I can answer very, very briefly and more than just my hypothesis of why specific gravity is better. But I think, so take this for what it is like it's not exactly like matrix effect in MS, but like the amount of metabolite that actually precipitates that then becomes part of my input sample is influenced by the other molecules that are there. And so we think specific gravity, because it's like the true density measurement of the sample compared to water, best relays that information.
Great, thank you very much, Elizabeth. Our last lecture will be given by Professor Robert Powers from University of Nebraska-Lincoln. Bob, this is PhD from Purdue and postdoc at NIH and IDDK. Right now, he's a professor and the director of System Biology Core, Department of Chemistry, and his talk is titled right here. Please, Bob.
Yep. Thanks, Leo, and thanks for the rest of the members of this workshop for coming forward today and showing you the capabilities and promises of metabolomics. I'm going to kind of full on in terms of what Liz is doing at the very end and try to highlight some things we need to consider to make sure that we keep on getting good productive outcomes from our work. And I think one of the challenges that we face in terms of metabolomics as a field is that it looks really easy to do. Just grab some biological samples, throw it in the NMR, mass spec, take that data out, put it through some software, and lo and behold, you have all these biological differences and so forth. But as most of us well know, as we start delving into the details, there are a lot of steps, a lot of aspects we have to need to do, and that every one of those steps is a possible source of problem in terms of errors. And if we think in terms of broadly, what some of these problems are going to face is that we're dealing with the metabolome itself, which is inherently unstable. Most of us are not trained biostatisticians, yet we're using high-end statistical techniques to analyze our particular data. We're not identifying, detecting all the metabolites that are actually present. And I think this kind of gets wrapped up in the fact that as a community, I don't think we have well-established, agreed upon best practices and protocols for us to go ahead and proceed. So how does this manifest itself? And again, without the presence of best practices and challenges, we kind of do what we do in our own labs, and that differs from everybody else. And when we start comparing, we start running into problems. And so we did a meta-analysis of about 24 different metabolomic studies looking at biomarkers for pancreatic cancer. So these all worked. They should have all identified the set. 24 is a significantly larger number of studies there. And so we went ahead and compared these, primarily looking at what they identified. Overall, there's about 655 metabolites that were identified by these 24 studies. Here's a heat map across, and we see no clustering based on analytical technique, sample type, and so forth. And if we look at further, we see roughly about 80%, 87% of these metabolites are singletons, identified by one particular study. And this is across both mass spec and NMR studies. I think there's only one that was using both of these. And of course, when we delve into the details, there's a lot of differences in terms of how these studies were done, what criteria they used in terms of fold change differences, what criteria they used for statistical significance in terms of p-value, whether or not they did any type of multiple hypothesis corrections, how many samples did they present, how do they analyze the data in terms of qualitative, quantitative, and all these factors come into play. Makes matters worse that the pie chart in the bottom there is even when we identify metabolites that were identified across studies, more often than not, they were showing opposite trends. So one study there an increase in metabolite, and the other study there a decreasing metabolite. We made this plot and thought, oh, maybe this might tell us something. We see a lot of noise when there is small cohorts in terms of the number of metabolites that are identified, but then it seemed to kind of flatten out when there is larger cohorts. But then when we looked at what the actual criteria that those larger cohorts used in terms of the p-values for statistical significance, and A lot of them used a p-value of 0.1 or 0.3 in order to get some metabolites that were statistically significant, and that's probably why those numbers got flattened out in terms of smaller there. It's not all bad news. If we delve in and try to find some subset of metabolites, there is some level of consistency. Some metabolites are identified by more than one study. In some cases, they are showing the same general trend, but not all the time. But again, we're looking at the best cases being identified in less than 50% of the actual papers and still showing variations. If we start looking at some other higher criteria in terms of filtering down, we start seeing maybe a list here about 10 metabolites that might be real, that might be something in terms of associated with it. So there's hope in the noise is what I think, right? So again, this comes back to, at least from my perspective, that we don't have well-established best practices. All those studies use very different approaches, and that's the challenges. And again, the metabolome itself is very unstable. Statistics can be easily used incorrectly. We're not identifying everything that's there. And again, so some things to consider in terms of moving forward. We have a collection of different statistical tools that we're going to apply. And sometimes we go searching for the one that works. And what this summarizes is that if there's not an underlying difference in your data sets, you're not going to find that. So what you see on the left there is a PLS plot of random noise showing great group separation. And we've demonstrated similarly, if you look in the center, there is a trend there of PCA and OPLS. We're adding noise. The PCA behaves as you expect. As you get more and more noise, you get less and less separation. But the OPLS model shows great group separation, even though it's mostly noise. And if you compare, which is what those correlation plots are, there's no similarity between the actual features that are identified as group separation in the OPLS models with increasing noise. So at the end, you'd be just identifying random peaks as being your feature separation. So we have to be careful in terms of identifying the proper statistical models and properly validating that. And validation goes well beyond just reporting R-squared and Q-squared values. Those are really measures of the quality of the models and not really reporting the validation. We need to do something like permutation tests, CV ANOVA, report a particular p-value. Again, the OPLS model to the right here looks like perfect separation, but from the validation, it completely fails because it's all noise there. Right? Another thing to consider, as I pointed out, how many samples should we do? As Liz pointed out and others, we have this classic P versus N problem, right? We have very few replicates and a lot of signals. And so just a back-of-the-envelope statistical analysis, random choice is going to be likely that I'm going to have a couple of features that just trend coherently from one group to the other. That's going to give me a variance if my number of replicates are only like 3 or 4 or 5, right? If I have 1,000 or 10,000 mass spectra feature or a couple hundred NMR feature, randomly there's going to be a likelihood in terms of they're just showing the same trend. So we need to have a minimum number of biological replicates, somewhere in the neighborhood of about 10 to 20 or so. When we get to clinical studies, again, standard back-of-the-envelope power analysis suggests minimum number about 60 patients per cohort in order to get any type of statistical significance. And if you saw a lot of those pancreatic cancer studies, most of them were not at that particular level. They were barely at 60 in total. Right? Another thing we have to do, it should be a requirement, that we have to both normalize, as discussed previously, and also have to scale our data. If you don't normalize, you get meaningless results. Again, identified here is what unnormalized versus normalized data looks like. Same way in terms of scaling. But of course, we have a lot of normalization techniques to go ahead and choose. So which one do you actually choose? The good news is that most of them, except for the histogram approach, stay away from that one. It actually does not work. Will do well under normal circumstances when you don't have when you're not really stressing your data. But when you get to very small group separation or a very large group variation, there's a couple of techniques that actually stand out. Normalization does, but again, depending which normalization technique you apply, you will get very different models and very different interpretation of your data. So you got to keep that in mind too in terms of your choice. Normalization does well in terms of capturing the differences in terms of peak intensities or dilution factors, but it doesn't recover from noise. Noise, I think, is really one of our Achilles heels in terms of this field, in terms of statistical analysis. What this shows here is that as soon as you get to roughly about a noise of around 20%, we do not recover what the actual true spectrum looks like, regardless what normalization technique is, right? That's roughly 20% noise, signal to noise of 5 to 1. So basically, you're looking at a peak that less than that, you're probably not recovering its true variation across the data set, even by normalization, right? Again, this is a simulated data set with nearly ideal signal to noise, and even that noise level results in very large group variation. So noise is a real problem. So removing noise at all points, as I think as Dave was indicating, would be a great bone if we can completely eliminate the noise in terms of data analysis. Again, real data scaled by the signal to noise. We can see the group variation scales proportional to the signal to noise, or how many scans we collect in terms of the NMR. Again, the only difference here is the signal to noise in terms of the actual spectra, in terms of that impact. Liz pointed out earlier, we've done a number of projects together. One of the things that we've recently done is showing we can get some improvement by using non-uniform sampling and also increasing the cycle time by using parametric relaxation. Again, anything that reduces your noise is a good thing in terms of removing that in terms of the spectra. Again, keep in mind that all the statistical tools they're not the same, right? They have particular application, particular design. Quick summary here in terms of PCA, robust tool we use all the time. It's primarily looking in terms of identifying the largest variance in their data set, regardless of what the source of that is. We routinely do PCA to first validate that there is really an unbiased difference in our data set. And then we apply an OPLS to go ahead and identify what the actual features that contribute to that. We tend to avoid PLS because that tends to mix variables across multiple different components, which is kind of summarized here again. So if you see interactions when you're analyzing a PCA data and trying to identify what the actual major features are contributing to group differences by something like a biplot, well, that is problematic because each of the PCAs are a mixture of all the variables, all the peaks and so forth, and so across the entire spectrum. So it's difficult to kind of pull that out. Whereas OPLS is actually designed to actually put in all the features that are associated with the group separation in the primary component. So when you pull something out like a backscaled loadings, regardless of NMR mass spec data, all those variances are directly due to their primary group differences. So again, it's important to understand what the underlying goal is of the underlying statistical technique. So to further address that, we kind of did a simulated analysis with some data set, looked at 5 very common statistical techniques made to as shown here on your right is what the actual variation in terms of the data was. You can see at the very beginning, it's a lot of noisy, very few peaks that differentiate, and then it gets very clean. The good news is that when we compare the easy, the high group separation, all the statistical models, for the most part, did very equivalent well in terms of regenerating those variants, telling us what the major features were, telling us what the group differences. When we really push things, and we had very small group differences, very large within group variance, that's when we started seeing significant differences in the performances of those 5 different techniques. And in our opinion, in this data set, OPLS turned out to be better at that extreme case versus the other 4 techniques. The other issue that we're facing is that there's a lot of dark matter in terms of the metabolome, right? Whether or not we're using NMR or mass spec, we're typically only seeing a few hundred out of the tens, if not hundreds of thousands of metabolites that are there. So the way that I think we need to move forward is being technique agnostic, right? We really need to be looking at applying multiple analytical techniques to get a broader coverage of the actual metabolome. And again, this further illustrates both the challenges we see with both my NMR and mass spec, right? We have a very small molecular weight window in terms of where most metabolites fall. And there's a very limited chemical shift dispersion in both 1D and 2D in terms of where most metabolites fall. So differentiating between them all can be very difficult if you use one T and the other. And this just highlights the benefit, the strength of combining both NMR and mass spec. This is a study recently done where we first started off with the GCMS data analysis, and we were getting kind of spot coverage. We came in, did the NMR, and we filled in all the holes. NMR detected almost all the missing metabolites from this metabolic pathway, showing us the entire process. And there are some occurrences where we detected the same metabolite by both NMR and mass spec, and with no standardization, no normalization, generally those metabolites had at least some similarity in terms of the relative trends between the two different techniques. So we were able to go ahead and fill that in. Now, more often what we're doing in my group is applying a multi-omics approach. One of my students actually approached me when she finished a particular project and said, I completely got a different view of the system when I looked at first metabolomics data, then when I looked at the lipidomics data, and then when I looked at the proteomics data. All three data sets tell me something completely different. It was only when I put all the data sets together that it actually makes sense in terms of what was going on. And in this case, we were trying to identify what the actual lethal target of a novel new drug in terms of treat TB. And we set up a parallel process where we were able to use the same set of samples to go ahead and generate metabolomics, lipidomics, and proteomics data source from the actual same sample set. And there's a lot of data there, but that massive data is self-correcting. So we were able to go ahead and take all the major protein changes, all the major lipid changes, all the major metabolite changes, and generate one coherent correlation map, which is shown here. And that lets us identify the subset of those molecules that are all correlated to the actual response. I have a poster, you can come back and get more details. And then with the identifying which ones are positive and negative correlated, right, then we can go ahead and set up a network connectivity map. This obviously is not very pretty, it's not publication quality, but the student can manually convert that into a very traditional kind of cartoon, showing again all the key lipids, metabolites, and proteins that are all self-correlated and responding to the treatment of the drug and generate one overall uniform response, okay? So I'm running out of time here. We're talking about annotations. One of the things that we're promoting is using pooled samples from different groups and collecting the standard HSQC, HSQC toxi, and HMBC. And we developed, modified the pulse sequence so we can kind of do a decoupled HMBC. HMBC, Art was showing earlier about the value in terms of C13 label, C13 probe, in terms of doing C13 correlation. I have a simpler approach. You collect an HMBC, you do a correlation with that experiment, and you get the C13 correlation from that particular experiment. And obviously, it's much easier to collect an HMBC and higher signal to noise, and you can go ahead and create that and generate those graph maps and so forth. NMR, we have a bunch of nuclei. Why not use all the nuclei? So this is showing a protocol to go ahead and collect phosphorus, nitrogen, and carbon spectra from the same sample. And of course, we can go ahead and get really, very nice samples if we label by C13 and N15 for cell extracts. Liz already pointed out, chemical shifts, perturbations is our big problem in terms of automation, so we need to know what really are the range of chemical shift variation so that we can go ahead and then put that into an automated matching in terms of the reference data set. So this is a truly automated approach, no person intervention. It will actually adjust for the chemical shift variation and distances and meet and match those, and we can kind of get fairly good results. But I think, as Liz pointed out, which I completely agree, when you look at trying to fit 1D NMR, There's so many peaks, so many possibilities. As soon as you have a database that becomes larger and larger, the false positive becomes larger. So I think the approach like David is doing, where you have a defined database of metabolites that you know that are present in the biofluid and you fit for those is probably the best way in terms of moving forward. So with that being said, I'll leave you with a list of things that we need to think about and address as a community in terms of looking at in terms of deciding on best practices, standardizing how we're processing our pipeline, choice of statistical approaches and methods, and so forth. So thank you very much, and I'll be happy to answer any questions.
Great, thanks, Bob. Any question for Bob and also for the panel, if we still have some time? Great, here one.
Thanks. Hi. Because you talked about the importance of normalization and that there's lots of different techniques. Do you have a preference or a go-to?
Yeah, so our preference is probably doing the PQ type of normalization. I think that's what our routine approach is. But I don't have strong, definitive mathematical, logical explanation in terms of, besides that simulation in terms of being a relatively robust performance, in terms of why that should be better versus any other type of approach. But I think we have to be careful and realize that everything that we do to our data set impacts in terms of how we're going to interpret the results at the end. And we can give ourselves false view that something's going on.
Okay, thank you.
Any other questions? Any? Okay.
Oh, okay, we're live. Okay, great. It's maybe more philosophical, so sorry to put you on the spot, but all of the efforts that a lot of us are doing is to make this more and more automated and low touch. Do you think that will introduce any potential negative consequences?
I think the possibility is always there, right? The more things are automated, the more it's easier to kind of go ahead and break the system. But I also think the positive is that if we think about it hard and we have what we know is a relatively robust approach, and it's automated and it guarantees that's what's being done over and over again, then I think that would probably be the positive outcome, right? So I'm more positive than negative, even though I tend to be a negative person.
You are? But that's a very important topic you point out. Any other questions? If not, I think the panel will stay here for a little while, so you can come here to individually ask questions if you have. And let's thank the panel again. Okay. I just want to make a very quick announcement. This is the workshop I'm working with International Society of Magnetic Resonance in Medicine. We are preparing metabolomics and the metabolomic imaging workshop that will be the end of next year, very likely. So just keep an eye on that information. Any other question? Anyone want to make comments? Anything? If not, thank you very much, and really appreciate everyone here. Okay? Yeah. Free gift here.